Here's the pinnacle of neurodevelopment, a little organoid. She made her own brain all by herself with all the ventral and dorsal patterning centers and radial glia migration and cortical folding and proliferation. She still has more to do, but she's done perfect so far. Say hello, Genevieve. Hello. 2 days old. Lots to learn. But she knows how to drink milk. Alright, so today is the day that we're going to peer into the nervous system and visualize all these incredible and fascinating structures. It really is astonishing. And so I want to start at the very beginning. We talked a little bit last week about the first neurohistology. If you look at these images here, you can see this hippocampal fold. This was the first time the hippocampus had ever been visualized. And see if you can guess who did these images. You can see the one on the left is a little more simplistic than the one on the right, but they were both done in the 1800s. The first one was done by Golgi, that's the one on the left, and then a few years later, Ramon y Cajal improved the Golgi stain and drew this hippocampus in a little more detail. And these were the very beginnings of starting to understand that neural networks existed in the brain and what a neuron even was and what a synapse even was. And it was from this work that we elucidated how the brain functions, that it's not just a mass of oatmeal, but this complex interwoven network of signals through these neurons and synapses. And because of that, Ramon y Cajal and Golgi together won the Nobel Prize. This then set the stage for many scientists and doctors and other people who would come along later and investigate this further. And it's pretty fascinating to see that even in the 1920s, 1930s, we had this incredible detailed understanding of neuroanatomy and neuropathology way before we knew what DNA or proteins were for. We had already constructed these maps of the nervous system, and many people had made these very artistic and detailed drawings of neuroanatomy. So I've just put some up here from some books that I happen to own, and you can see the detailed networks, you can see detailed cell pathology, and these are both, these books that I've shown here are from the 1920s and 1930s. Now, they may not have known all the reasons that they saw the things that they did, and certainly they didn't know the molecular biology yet, but even with their crude tools, they were able to take meticulous observations and link these sort of histopathologies that they were seeing to the clinical syndromes that they were dealing with. And so even back in the day, they were seeing gliosis with traumatic injury, for example, or neuroglial fibrils with dementia patients. So these sort of links and observations over many years through the tremendous effort of these scientists is how we came to know what we know today. So I think it's useful to start with just sort of a quick review of the stains which we will use in this course. So you don't need to know any stains outside of what's listed here. Now, of course, there are many, many others not listed here, especially once you start getting into very specific stains like antibody stains that can target specific molecules. But these are generalized stains, and they're quite common, especially in neuroscience. And so I think it's useful just to cover these and know these ones in depth. So the first one is the hematoxylin and eosin stain. This is used in all of pathology. It's quite common, probably the most common one used. And like most dyes, it helps to have more than just one dye. If you just stain one thing, it doesn't really give you contrast within the tissue. So typically we'll use 2 dyes at the same time. This one, the hematoxylin, stains nucleic acids purple and it stains proteins pink. So the nuclei tend to turn purple, so you know where the cell bodies are, and the proteins, which are ubiquitous, there's everything from intracellular proteins to extracellular matrix. These all turn a pinkish color to some degree or another. So I'll show examples of these at the end, just so that you can get a sense of what they actually look like. But let me run through the wording here. So the Luxol-Fast blue, it is a stain that targets any fatty substance, especially myelin. So this is useful in the nervous system when we want to look at myelinated tracts. And we usually use this with a counter stain or a secondary stain like Crescel violet, and that way you can see the Crescel violet bound to neurons and see the tracks that come from those neurons as a separate color. So the Crescel violet is kind of like the hematoxylin. It binds to nucleic acids. This one tends to bind RNA really well also, and neurons have lots of RNA, especially like at the synapse and things, so you tend to visualize the neurites a little bit better. Okay, now a totally different type of stain is the silver stain, and the one that we use mostly in this class is a Bielczowski stain. Bielczowski, or a reduced silver stain. There's many actually. There's many variations of a reduced silver stain, but the silver stain targets the cytoskeleton, specifically microtubules and neurofilaments or intermediate filaments. And this is really good for visualizing these sort of architectures of neurons. Glial cells don't tend to have as much cytocellular architecture, but the neurons have a lot. So you can see a lot of things, even neurofibrillary tangles, neuritic plaques, and Alzheimer's, things like that. Now, the original silver stain was actually by Golgi. He was using photographic dyes, and back then photography was done with silver nitrate, and that's what enabled black and white photography. And when it combines with potassium dichromate, you may remember from your inorganic chemistry, forms silver chromate crystals. And for some reason, these tend to only form in a small percentage of cells. And that's really lucky, because if they didn't do that, then you would essentially get a giant black plate of your dye. But in this case, when they just tend to form in a few of the cells, maybe it's micro tears in the membrane of the neurons, but by doing that, they form in only a few of the neurons, and that enables us to visualize individual neurons, not a bunch of overlapped stained neurons, but individual neurons. And as you sketch this, you can kind of combine different slices so that these few neurons that happen to stain with the silver stain, you can kind of put it all together in a single drawing and make a really nice image. So Ramon y Cajal improved that silver stain a few years after Golgi, like I talked about. Now here's an example of each of those stains, just so you can see it. Don't worry about identifying anything here. We'll talk about all this later, but this at least lets you get a sense of what each dye and dye combination look like. Now normally in lab we would use the microscopes to look at these neural tissues under the scope directly, but in order to forego that this semester, I'm just going to show the slides here. So you won't have as much time to explore and kind of search around all these structures, but it is a little more formal and streamlined, so at least that's better in that sense. So for the exam, you only have to identify the things that I've listed here. If it's not listed here, I'm not going to ask you to identify it. So it might seem kind of easy at first, but today's lab, you really should take the time to make sure you can identify these structures, not just on one image, but possibly from multiple different slices or different stains. So think about that as you go through these, but there's nothing due today. All you have to do is take the time to study these histologies. Okay, so I'm just going to walk through them. First, we're going to look at sort of the macroscopic histology. Under the microscope, you would not be able to see an entire spinal cord cross-section all at once. You're only going to be able to see small regions of it. So I'm going to show the macroscopic section here, and then we'll look more at the microscopic sections with the stains later. You should note on these macroscopic slides, it's kind of funny because of the reduced silver stain, the gray matter actually looks lighter and the white matter actually looks darker, so don't get confused with that. So we'll do the spinal cord cross-section first. The gray matter, of course, is the central classic H-shaped sort of region here. The white matter is all the white matter tracks, the ascending and descending tracks. The central canal is located right in the center. It's not that opening off to the left that looks like an opening. That's actually a vascular canal, but the central canal is lined by a remnant of the ependymal cells, so it actually has this sort of epithelium surrounding it, which appears dark with the reduced silver stain. Just anterior to the central canal, we have the anterior white commissure, which is just fibers that are crossing over. And then at the dorsal side of the spinal cord, we have the dorsal rootlets, which is where the sensory information comes in. At the anterior or ventral side of the spinal cord, we have the ventral rootlets, which is where motor information goes out from the alpha motor neurons, which we'll look at in a second. The motor neurons themselves live in the ventral horn of the gray matter, and you'll see as we zoom in on them, it no longer appears gray or white. It actually becomes more of a texture issue. The coloration really disappears, so don't rely on that to identify where you are. What really defines these is that the white matter is white because of the myelination, and the gray matter is gray because of the neuron bodies. So when you see the alpha motor neurons up close, they're just going to look like big neurons, not gray neurons per se. When you look at the white matter regions, for example, the dorsal columns where sensory information is carried up to the brain, we can see myelinated axons, and then this entire spinal cord is surrounded by meninges. At the anterior side, again, we have the anterior median fissure, which is a true fissure, sometimes called a sulcus, but it is separated quite a bit, and there's usually a vessel that will run up and down in there. It's useful to note that in the central nervous system, typically meninges and vessels tend to run together. So usually there'll be some meninges and vessels that run in that anterior median fissure. You can also see a vessel that's sort of flattened but large at the anterior side of the spinal cord, the lower side of the page. And that is the anterior spinal artery, which feeds blood supply to the spinal cord. And that's often fed, at least at the lower regions, by the artery of Adamkiewicz, which comes in off the aorta and feeds the lower portion of the spinal cord. So heart surgeons, for example, have to worry about this. If you clamp off the aorta, you can cause a stroke in the spinal cord. So just useful connections to make there. Finally, at the posterior side, you can see the little sulcus between the dorsal columns. Moving on to the peripheral nerve, the structure here is pretty basic. An endoneurium refers to the myelination around a single axon, and then you have a bundle of axons which are surrounded by a perineurium. That structure, the bundle itself, is called a fascicle. You can actually have fascicles within fascicles, as you'll see on the histology, and then all the fascicles together is surrounded by an epineurium. The perineurium tends to be a very fibrous structure, which you'll see, whereas the epineurium tends to be more loose, cloudy structure, at least on the histology. And of course, there are vessels that run in and out of here to supply energy to these cells. From last week, you'll probably recognize that the image here is a 3D image from a scanning electron microscope showing a transection of a peripheral nerve, so you can see the vessels as well as the nerves themselves. The vessels can run in the fascicle or between fascicles. There's no specificity there. Oh, and I forgot to mention that if you have access to the lab or happen to be in there for other classes, you are welcome to look at these slides, pull out a microscope, plug it in, hook it to your computer, and use the Capture OEM software to take pictures. The slides themselves are in the blue boxes, but all the images that you need to do well on the exam are here. Okay, we're also going to look at the peripheral nerve with the fibers separated, what are called teased nerve fibers. So if you look at the structure of the spinal cord, when the rootlets come out and join together, then you have the joining of the peripheral nerve, and this contains both motor and sensory branches, and most of them are myelinated. You may see a few that are not myelinated, which will be, roughly a third of the diameter of the other fibers, but most of them will be thick fibers, and you may even get lucky and see a node of Ranvier. Looking now at the cerebrum and cerebellum, we'll look at the cerebellum first. There are many similarities but many differences here, and mostly differences. So the cerebellum only has really what we classify as 3 layers of the cerebellar cortex. You'll see that the cerebral cortex, what we normally think of as the brain cortex, has 6 layers. But here in the cerebellum there's only 3, and they're pretty easy to identify. In the most superficial layer, we have the molecular layer, and that's called that because it's very sparse. You can see there are very few neuron bodies in there. The dark bodies, quite sparse, as I said. The next layer down is only one cell thick. It's the big round Purkinje cells that you see below the molecular layer. They sort of line the bottom of the layer. And then the next layer is the granular layer. And you can think of this as the grains of sand. It's granular because there are just billions and billions of neurons that are small and round underneath the Purkinje layer. And these neurons are why the cerebellum has nearly as many neurons as the cerebrum. And then all of this has to have some sort of input and output, so that's what the white matter is. You can see a subtle change to a lighter brown below the granular layer. If you're up close, you can actually see that there is a fibrous sort of direction to the white matter layer. The classic circuitry here is really interesting. You don't need to know this for the exam, but it's just worth pointing out that when we look at the pons, we're going to see some pontine neurons. Those get input from the cerebral cortex and then project their fibers up to the cerebellum through these fibers called mossy fibers. These mossy fibers come in, synapse on all these granular neurons, and then those granular neurons project up to these cells in the molecular layer. And they do this by these fibers called parallel fibers that are mostly in two dimensions, these sort of T-shaped fibers. And then these few neurons in the molecular layer then send their projections back down to Purkinje cells. And then the Purkinje cells are the main output of the cerebellum. So there's obviously more complex circuitries as well, but that's sort of the canonical circuitry of the cerebellum. So let's compare this to the cerebral cortex. Okay, in the cerebrum, we have typically 6 layers of neurons. The hippocampus is an exception to that, but you can see here in this photograph of the cortex, at the most superficial layer, which is labeled F, it looks a little confusing, but that's a sulcus in the brain that you're seeing at the top right of the image, and then the most superficial surface layer of the cortex is F, which is the least dense in terms of neurons. It's the most sparse. Just like the cerebellar layer, it has very few neurons. And below that, you can see a dense collection of neurons through all the layers for layers 2 through 6. So this is the result of sort of a vestige of early development. There are these cells called Cajal-Retzius cells, for example, that migrate to the surface of the brain, essentially defining the surface of the brain, and they secrete factors like relin and other chemicals that help the neurons, the neural precursors, migrate up in this inside-out sort of pattern that will ultimately form all 6 layers of the human cortex. But because of that, the first layer, this layer 1, tends to remain quite sparse. Now, I'm not going to have you have to know layers 2 through 6. If you're an electrophysiologist or something, you will get to know those layers intimately, but for our purposes here, you only have to define layer 1 or layers 2 through 6. Now, there are some defining features. For example, layers 3 and 5 tend to have the most pyramidal cells, which we'll look at in a minute. But all layers, there can be pyramidal cells in other layers as well, but there are a lot of distinguishing features once you start looking at individual cells in each layer, and thicknesses of layer, it turns out. So depending where you are in the brain, you can expect to find different thicknesses of each layer, and this has to do with the canonical circuitry of the cerebral cortex. So classically, information, and like sensory information, will come in to layer 4, and then it gets processed up in layers 2 and 3, and then it gets spit back out of layers 5 and 6. So in cortices like the visual cortex, where you have a lot of sensory input, you can expect to find a very thick layer 4. And in the motor cortex, where you have a lot of output from the cortex, you can expect to find a thick layers 5 and 6. And then in cortices where you have a lot of processing between multiple inputs of information, you can expect to find thick layers 2 and 3, like the prefrontal association cortex. Here's an image of a pyramidal cell in the cerebral cortex. This is showing both the histology and also a drawing just to help you identify that the dendrites are much more prolific than what you see just in the histology. And typically an electrophysiologist who's searching for these specific cells will light them up with some sort of intracellular dye. But in the histology, we can see lots of fibers coming out, but you can't necessarily tell which fibers come from which cell. So the pyramidal cells are called that because they look like pyramids, and it's worth noting that the axon itself is pointing downwards, and the dendrites are all pointing up towards the surface. They always point towards the surface of the brain. This image with the mouse is just showing the classic circuitry again, a whisker sensation going into layer 4 from the thalamus, and then getting processed and reacted to with some sort of motor movement out of layers 5 and 6. Alright, another incredibly important structure in the brain is the choroid plexus, and this is often overlooked and neglected, but it turns out to have really vital clinical applications. So you can see in the image here, we have a ventricular system with the lateral ventricles and the third and fourth ventricle, and also a central canal, which is basically a remnant of the neural tube folding in early development. In the spinal cord, this typically closes off. This is the central canal that we talked about earlier, which we still haven't seen on the histology, but I'll point out to you. And you'll see it's typically quite small, but if you have a traumatic spinal cord injury, for example, this can open up again and become what's called a syrinx, a sort of pressurized fluid canal. Now, looking back at this ventricular system in the brain, you can see choroid plexus in red, which lines all the ventricles. Some people don't recognize that choroid plexus does indeed occupy all the ventricles, and it produces cerebral spinal fluid. It's sort of this spongy substance, and it can be quite friable, especially in neonates. It turns out that if a baby is born quite prematurely, its choroid plexus is quite susceptible to reactive oxygen species and oxygen damage, and they tend to hemorrhage. And you can imagine that the problem with this is that the hemorrhage will then occupy the space of the ventricles, and that creates a problem because the ventricles cannot empty the cerebral spinal fluid, which exits down by the fourth ventricle and then surrounds the brain. So instead, the fluid, the cerebral spinal fluid, will fill up inside the brain, expanding and expanding until it pressurizes the brain and creates a condition called hydrocephalus. You'll see if you work in pediatrics or with a pediatric neurosurgeon that this is a condition you'll see frequently. And so there's a couple things you can do. In older children, you can put in a shunt and try to bypass that cerebral spinal fluid either into the abdomen or even into the vascular system. But in very premature babies, the head is tiny, and the shunts and the machinery and everything is quite large, and so it's not feasible to do this in early neonates. When I was in residency, I would typically start each morning at 4 a.m. and go up to the neonatal ICU and use a syringe and needle to withdraw this fluid manually from the ventricles of these tiny little infants, and it's one of the saddest things that you can witness. Fortunately, they tend to do well if the hydrocephalus is controlled and there's no other comorbidities, but there's a lot of potential for development of better interventions and better devices, I think. So if that's something you're interested in, it's definitely worth pursuing. You can see that this choroid plexus looks spongy even on these electron micrographs. Remember that there's no color on electron micrographs, so that's an artificial color. The color there looks like ice cream cones for some reason, but this spongy material also tends to clog up shunts really, really well. So another thing that you'll see in pediatric neurosurgery is that patients will come in with headaches who already have a shunt, and the question is whether it's a shunt failure and they're getting hydrocephalus again, whether it's just a random headache, and typically if it is a shunt failure, it's caused by the choroid plexus sloughing off these sort of protein materials that clog the shunts. So again, a lot of innovation is waiting to be made in those devices. Now, if you were to slice through the choroid plexus, you would see this sort of villus structure. Inside the choroid, you have these vessels, artery and vein, and that's what supplies the fluid to make the cerebral spinal fluid. The artery tends to have a very thick wall. It's the tunica media, tends to be muscular and thick. That's how you can define an artery versus a vein. And you can see in both artery and vein, typically there are red blood cells, which are these anuclear, reddish little round things that are about 8 microns in diameter. If it has a nucleus, it's a white blood cell. Now, there's an epithelium that lines all the connective tissue. That is the choroid plexus epithelial cells. Those are the actual cells that produce the cerebral spinal fluid, and they're connected by tight junctions. And these epithelial cells will ultimately link up with the ventricular ependymal cells, which is also the epithelial cells of the ventricle, which we'll see in a little bit. Now, the last macroscopic histology that we'll look at is from the brainstem, the dreaded brainstem. And what I want to do here, the brainstem can be drastically overwhelming, but I don't want you to panic. What we're going to do is just cover some very basic components and kind of let you build a shelf, build an architecture to stack future information on. But if I just overwhelm you with a bunch of things, none of it's going to stick, and then it won't be that helpful to you. So let's just learn a few things, but learn them well. So in the medulla, you can see that at low magnification, we can see the pyramids, which are the cortical spinal tract at the base of the image here, and that is anterior. Then we have also the medial lemniscus, which is where sensory information rises up through the brainstem and goes back to the brain. So all you have to know here are the descending motor tracts and the ascending sensory tracts. We're not going to learn all the complex spinocerebellar and the spinothalamic tracts and all that. Let's just focus on basic sensation through the medial lemniscus. You'll remember the pathway is actually called the dorsal column medial lemniscus pathway. So we're going to look here at the medial lemniscus and see its unique structure in the medulla. Also, we have the inferior olivary nucleus, and I just included this because it's so obvious. It's this big wormy thing. And on here, it just looks kind of light colored. And you'll remember, as in the spinal cord, when something was light colored on these particular stainings, that typically means that it has neuron bodies in it. And that's the case here. That's why it's called a nucleus. From far away, it just looks like a big worm. But when we get close, you'll see that it's not a worm at all. It actually is distinctly filled with large neuron bodies. Also, just to keep our directions straight, let's make sure we know where the fourth ventricle is. It's at the back or the top of the image, the posterior side of the medulla, and it is lined with those ependymal cells that I talked about, and we'll see that very distinctly on the higher magnification. And then also, just generically, we should know that this is surrounded by meninges and blood vessels. On these slides, most of the meninges and blood vessels have been removed, so you won't see them necessarily, but it's just something to keep in the back of your head. All right, at higher magnification, we kind of already talked about this, but you'll have to know nuclei versus tracts. So nuclei, for example, the inferior olivary nucleus, it's filled with big neuron bodies, versus tracts, which won't have any neuron bodies. They'll have little glial cells, but they won't have big neuron bodies, so they're pretty easy to differentiate. Also, the tracts will tend to stain with the Leuxal-Fast blue color, whereas the nuclei will tend to stain with the Crescel violet color. And as far as ascending and descending fibers, you cannot tell that from the stain. You can only know that by knowing your neuroanatomy, but I'm not going to quiz you on that. It's just a general term to keep in mind as you look at these images. Commissural fibers, of course, are neither ascending or descending. They're going to be crisscrossing through the tissue. And then we already talked about neurons, glia, and ependymal cells. Great. Okay. Now, going on to the pons, if we look at this again, it can look really confusing and intimidating, but don't let it be that way. Let's just establish the basics here. So low magnification, you can see a trigeminal nerve, a giant nerve coming out laterally on both sides here. This will take us back to the trigeminal nucleus. Now, the trigeminal sensory nucleus is kind of funny. It actually runs up and down quite a bit through the brainstem. But there is one tiny little motor component of the trigeminal nerve, which you may remember goes to the muscles of mastication, and that's called the trigeminal motor nucleus. And that will be right at the posterior side where this fifth nerve, fifth cranial nerve goes back into the brainstem. So we will visualize that quite well. Now, just like the medulla, we will also see the fourth ventricle here at the back side, the posterior side, or the top of your image, which is again lined with ependymal cells. Now, the corticospinal axons here, they don't all stay in a singular bundle like they did in the medulla. In the medulla, we had the pyramids at the anterior side, but here we have sort of a separation of several bundles of the corticospinal tract, and it's sort of dispersed through other fibers within the pons. So we have these unique fibers that are decussating and crisscrossing, commiserating across the pons, and those are called pontocerebellar axons, whereas the corticospinal tract is running from head to toe. It's going up and down, and it is running through in between all those pontocerebellar axons, and so it looks more like these muscular bundles that have been transected. That is the collection of corticospinal tracts. If we were to zoom in again, we would be able to identify particular nuclei here, which I'll talk about a lot more once we see the microscopic histology, and then we've talked about everything else here. Oh, and it is worth pointing out that the reason there's a red box here is because the entire pons does not fit on your microscope slide, and so it's been cut in half and turned sideways. So when you don't see the entire pons on the slide, that's why. So again, I'm just putting up here the exact list of structures that you need to know. I will not quiz you on anything that's not on here, so it makes it pretty easy to study for, and we'll go through this again on the microscopic histology now. So again, this is just showing how you would set up the microscope and connect it to your computer. Let's start with the spinal cord slide. So the first thing we can see here is the very center of the spinal cord, and we see that this is all in a Bielchowski stain. It's just all brown, just different shades of brown. And at the very center there is the central canal lined by this dark sort of remnant of epithelium. It's not super obvious on this. It's been broken up in the tissue preparation. Some of the spinal cord sections will have a much more obvious one, but you get the idea here. At the lower portion of the image, that is the ventral fissure, which is quite apparent here, but you can't really tell because obviously we can't zoom around in this photograph to make sure, but you get the idea. You can see a small vessel and probably some pia mater running through the ventral sulcus here, ventral fissure. And then just anterior or in between this ventral fissure and the central canal, right in between, there's some fibers going laterally, horizontally across the page. That is the anterior commissure. So with this Bielchowski stain, you may be thinking, well, I can't tell what's a cell and what's a fiber, and that's okay. Like I said, every stain has strengths and weaknesses. The strengths of this are that we can see the cytoskeletal architecture quite well, but the weaknesses are that we don't really know where all the cells are. The glial cells actually don't have much cytoskeletal architecture at all, and so they don't show up very well, whereas the neurons do. If we scoot over just to the left on this slide, we'll run right into the lateral horn where the alpha motor neurons are, and these neurons are some of the biggest in the whole nervous system. They're really obvious, and during the staining process, they've actually been dehydrated and shrunken down, and because of that, they leave these halos around them, but their original size was even bigger. They filled up these halo spaces here. So, like I said, they're very large and obvious neurons. You can also see at the ventral or anterior side here down in the lower left that there are some dorsal roots. Those are the motor rootlets. And there are also some vessels and meninges running over the surface of the spinal cord here with a few little vessels plunging down into the spinal cord. And I've just outlined the whole ventral gray matter horn here just so you can kind of get an idea. You can see that it's not discolorated compared to the rest of the tissue. It's more of a texture change that will make it appear as though it's gray matter when you're looking at it from far away. Now if we move to the posterior side of the spinal cord at the dorsal root entry zone, we can see that this is where sensory neurons would be coming in, and we can see many small dark little dots. Again, they have halos. These are neurons, but these are interneurons, and so these are not nearly as big as the alpha motor neurons, and much harder to distinguish as neurons. if we zoom into 400 times magnification, previously we were only looking at 40 times magnification, but at 10 times that, we can see these nice neurons and little round circles, which are glial cells. And you can tell here that the glial cells don't really have a nice defined cytoskeletal architecture. Some of them you can see a little bit of a halo, but not much. So typically we only look at the glial cell nuclei and not the entire cell body itself, whereas the neuron, the cell body of the neuron is quite obvious. All right, let's jump over to the peripheral nerve now. So here's a fairly zoomed out image at 40 times magnification of the peripheral nerve. We can see a distinct collection of fascicles, as well as the epineurium, which is sort of softly holding them all together. And you can see many vessels within this peripheral nerve as well. So let's zoom in a little bit more here. Now we can see distinct fascicles with a tight band of sort of fibrous perineurium. And then within those, on some of these, if you zoom in far enough, you can actually see the individual axon, a hollow center surrounded by some pinkish myelin. And between different fascicles here, we also have some vessels, a large artery, for example, with many red blood cells and a couple white blood cells within it. And then at the periphery, you can see some epineurium, this sort of cloudy substance that contains all these bundles of fascicles. I think these cross-sectional sections make it pretty easy to identify things. If we look at a longitudinal section, though, it gets a little more tricky. Here you can see, again, some myelinated axons in the center, but off to the left, you can see that there's not many cell nuclei in that, and that's because the cell nuclei that are in that are fibroblasts, and they're quite sparse. They're just laying down new extracellular matrix to make the perineurium. So that left side bundle of acellular fibers, that is perineurium, that is the edge of the fascicle. Once we see all these cells with the hypercellularity here, with many cell nuclei in these fibers, you can tell that that's where the myelinated fibers start. So you might think, well, what are those cells? Well, they're not neurons, because the neuron bodies for these nerves will either be in, well, I should ask you, where would the cell bodies be? So think about that. Where would the sensory neuron bodies be, and where would the motor neuron bodies be? Obviously, we've already looked at the motor neuron bodies. They're in the ventral gray matter horn, and then the sensory neuron bodies, those would be in the dorsal root ganglion. So what cells are we seeing here? Well, it's the Schwann cells. It's the cells that myelinate these nerve fibers, these axons. And I've also outlined a little vessel there in red. So let's look at the dorsal root ganglion itself now. So at the top of this image is where the dorsal root ganglion would then attach to the spinal cord at the dorsal root entry zone. So all the fibers that are coming in from the bottom left, those are all sensory fibers, and they come up to these bipolar neurons, which look like little fried eggs. They're just this big bundle of cell bodies that are sensory neurons. If we zoom in closer, you can see why I called them fried eggs. So now if we tease apart these nerve fibers, we can actually start to see them individually. And because most of these are myelinated, they do appear quite thick, a few micrometers at least. And on this particular one, you can actually see a node of Ranvier, which I think is pretty cool. Moving on to the cerebral cortex here, we're going to see this is a Bilchowski stain. It's all brown, and you can barely see the surface up at the top right, and at the bottom left you can barely see that there's some white matter tract. So we've somehow captured the entire cortex in one photograph. That's usually pretty hard. Usually the human cortex is about 2 millimeters thick, so it can be quite difficult to get the whole thing in one image. Normally the top of a gyrus is sort of where it's thickest, and at the bottom of a sulcus is usually where it's thinnest. But even then it's hard to capture in one image at 40 times magnification like this is. So I've outlined just a few groups of pyramidal cells here. You can see that they tend to be around layers 3 and 5, but sometimes dispersed elsewhere, sometimes layers 2 or 6, things like that. And I've also outlined in purple the little veins and vessels that are going through the cortex here. Zooming in a little closer, we can actually see these pyramidal cells quite distinctly, as well as a large vessel that has some blood in it. If I zoom in close like this, you can actually see the individual pyramidal cells and their dendrites pointing upwards, and so in this manner you can actually tell where the surface of the brain is, even though you can't see it anywhere in this image. It's up to the top left. So moving to the cerebellar cortex, we've kind of already seen this, but let's take one more look at it. We can see this sort of wavy molecular layer, and it's worth pointing out here that unlike the cerebral cortex where we call things a gyrus or gyri, here we're going to call it a folium or folia. And along the base of this molecular layer we see many good Purkinje cells, and under those are the granular cells, and then most deep, although it's at the top left of the image mostly, you see these strands of white matter fibers. Zooming in gives us a really nice image that makes a distinct differentiation between these sparse molecular layer and the dense granular layer below the Purkinje cell. If we change over to an H&E stain, it looks pretty much exactly the same as the Bilchowski stain in this case. So really nice there. And then here we can see some vessels and meninges around the cerebellum as well. Next, let's look at the choroid plexus. So here we can see a similar image to what we saw before. We can see some large arteries, and usually there's some adjacent veins which are thin-walled, and the large wall, the large tunica media of the artery will make it nice and round, whereas the veins are much more oblong and collapsible. And between the vessels and the epithelium of the choroid plexus, we have connective tissue, and ultimately this epithelium produces the cerebral spinal fluid. So when you look at this image, the main thing you should know for the exam is just to be able to identify what is in each space. Is it blood? Is it connective tissue? Or is it cerebral spinal fluid? Okay, we saved the best for last, so let's jump into the medulla first. So if we just kind of zoom in to the center of the medulla here, we can see this interesting sort of what I like to call a rib cage structure. It kind of looks like a sternum down the center, and then off to each side, right and left, you have these sort of rib-like fibers. This is an identifying characteristic of the medial lemniscus. It looks just like this. But it's important to note that the true medial lemniscus pathway is what's coming in and out of the page or in and out of the screen. It's not the fibers that are crossing and look like ribs. It's the actual little axons, which are hard to see here, that are going in and out of the page, which ascend up to the brain carrying sensory information. The crossing fibers that you see are actually the olivocerebellar commissural fibers, which are basically arising from the olivary nucleus in the medulla and going to the cerebellum. So those olivocerebellar fibers are shown in blue, and the actual medial lemniscus pathway is shown in green. And then if we just scoot over a little bit, you can see that we arrive at the primary olivary nucleus, or inferior olivary nucleus. And here you can finally see that this is not just a big white wormy thing. It's actually quite pink on the H&E stain here, and it's filled with these large purple cell bodies, which are the neurons. So I've just sort of drawn a red line through the olivary nucleus structure, and then the green is outlining one of the neurons in the olivary nucleus, whereas I thought I'd just point out that all the little tiny round dots that you see above the olivary nucleus, what you see outlined in blue, is just one little glial cell nuclei. Then if we look down at the anterior side of the medulla, we can see that we have a giant corticospinal tract. This is the pyramid. Here you can't even capture the whole thing in a 40x magnification, which is the lowest we can go on the microscope. So I've just sort of outlined it in red here. This is the corticospinal tract in the medulla. And then let's look back at the posterior side of the medulla, which is at the top of your page here. And we can see here that the fourth ventricle is the space at the top, which is lined by these nice ependymal cells. Alright, last but not least, let's do the pons. So we have this big obvious structure that juts out from the pons. It's the fifth cranial nerve or trigeminal nerve, and it's really easy to find, really obvious. And if we follow that back, we will actually run into the trigeminal nucleus. You can't really see the big nucleus for the sensory portion of this nerve, which is huge, but because, like I said, it runs vertically up and down the brainstem, we're not going to capture a big portion of that in a horizontal slice. But what we do capture is a nice big portion of the motor component of the trigeminal nerve. And this is what innervates the muscles of mastication in the jaw. And so here we can see this nice circumscribed area of big neuron bodies. That is the trigeminal motor nucleus. If you look at these slides yourself, you'll find that sometimes these trigeminal nerve fibers, which look sort of bluish here, they go in and out of the slice, in and out of the page, or in and out of the screen, waving up and down as they go back. But if you just trust and keep following their directionality, you'll ultimately arrive near the posterior side of the pons and find this nice little obvious area of these big neuron bodies. So it's not too hard to find this trigeminal motor nucleus. Now if we keep scooting back, you can see that we arrive at the region of the fourth ventricle again. And again, it's lined with the ependymal epithelial cells. And you can see very distinctly on the right side of the image, when we're not in the fourth ventricle, there's no ependymal epithelial lining. But when we are in the fourth ventricle, as is seen at the top left of the page, there's these nice epithelial cells lining that fourth ventricle. Now if we move to the anterior side of the pons, again, just like the gross anatomy, it looks like a big pot belly here. It's a big round structure at the front. And here you can see a lot of sort of intermixing of different pathways. So this can get a little confusing, but again, don't panic. Just start trying to identify logically what these things must be. So we can see some fibers sort of running horizontally, crisscross left to right, and I've outlined some of those in green, or actually rather marked them in green. And then if you look at sort of these bundles of fibers, which is hard to see when we're so zoomed out here, but if you zoom in a little bit, you can see that the red are bundles of fibers going in and out of the page, and these are corticospinal tracts. Again, like I mentioned earlier, they've been separated out. And then lastly, we have something else here which we haven't really talked about, but that's these collections of big round cells. They have distinct large nuclei, and so the question is, what are these? Well, these are pontine neurons. These are neurons that live in the pons, and these are what are generating those green horizontal commissural fibers. Neurons arise from these pontine neurons, crisscross over to the inferior peduncle of the cerebellum, and these pontine neurons are getting inputs from the cortical neurons, for example. So when you see all these cells together, it looks like a bit of chaos at first, but you really can piece apart what these different cells must be. If it's a small round nuclei with no distinct cell body, again, it's probably a glial cell, and if it's in commissural fibers or descending or ascending pathways, then you know it's most likely oligodendrocyte that's myelinating those fibers, if it's in the central nervous system, or if it's in the peripheral nerve, what we would call a Schwann cell. And likewise, if you have a bunch of small little round glial cells within a nucleus of large neurons, then those are more likely to be astrocytes that support the neurons. Okay, so let's just do a little quiz here and make sure that we're all on the same page. So if I show you this image from the pons and tell you it's from the pons, and I should point out that I'm never going to put an image on an exam that's impossible to tell where it's from. There has to be some sort of clue where you are in the nervous system. Otherwise, you're just looking at a bunch of cells, and it's difficult. So there has to be some amount of clues. I'm not going to give you an impossible task here. So if I put this image up and tell you it's from the pons, and ask you what is A, you should be able to look at that and sort of see, well, okay, there's a couple things going on here. C looks like the big pontine neurons that we just talked about. B looks like axons going in and out of the page with a few little glial cells. And A looks like it has striations that crisscross horizontally through the page, or in this case vertically because the tissue is rotated, but the fibers are staying within this two-dimensional plane. So this suggests that A is the pontocerebellar fibers, whereas, like we said, C is the pontine neurons and B is the corticospinal tract. Alright, now let's see if we can take some of this histology and put it to clinical use. So let's do some clinical correlation here, and I'm going to do this with some classic board problems. This first one I actually did see in medical school, but most of these are sort of classic board questions. So the first one, 30-year-old woman looks over her shoulder to back up her truck. She was actually a semi-truck driver professionally, and she had the sudden onset of extreme dizziness and nausea. She comes to the ER and has a small pupil on the right and a large pupil on the left in a dark room. And the reason I say that there is because you never quite know which pupil is the abnormal one, actually. It could be the small one or the large one. But in this case, a dark room suggests that the pupil should be large, but she has this small pupil on the right side. She says that the right side of her face feels numb, and you test her sensation with pinprick and realize she feels no touch or pain on the left side of her body. There's no motor weakness, but there's some discoordination or ataxia in the arms and legs. So the question is, where is the lesion, and is it right side or left side? Okay, so I'll just admit, this is actually one of the harder problems here. So let's just go through the answers. Don't panic, this is not going to be on the exam, but I think it is useful just to kind of correlate what you're learning neuroanatomically to an actual clinical scenario. Maybe it'll help it stick better. If not, don't panic. So this is actually what's called a Wallenberg syndrome or a lateral medullary syndrome. So there I've just told you the answer. It's in the medulla, and this turns out to be the only sort of lesion, the only place in the central nervous system where you tend to have decreased pain sensation ipsilateral in the face, but contralateral in the rest of the body. So in this case, that means that the right side of her face is numb because the lesion is on the right side medulla. And that also explains the small pupil. What's happening there is that the descending sympathetic tract goes through the lateral medulla, and if it gets knocked out, then you lose sympathetic innervation to the eye. So it's worth noting that sympathetic innervation goes down through the medulla, out through the thoracic ganglia, follows the carotid up all the way to the ophthalmic artery, and innervates the dilatory muscles on the pupil. And so if you lose that, you're going to have a small pupil that cannot dilate. And then the reason that she's getting vertigo tends to be from knocking out the vestibular nuclei, and she also has a numb face because the trigeminal nerve has some sensory nucleus that runs inferiorly to the pons into the medulla, like I was saying earlier about the sensory trigeminal nucleus running up and down. And then with the small pupil, you actually get not just a small pupil, but the entire syndrome of a Horner syndrome, meiosis, ptosis, and anhidrosis, where you have a droopy eyelid because the Mueller's muscles are no longer receiving their sympathetic innervation and in the eyelid, and then also the anhidrosis because your sweat glands are innervated by the sympathetic nerve as well. So, and then the ataxia, that's probably explained by knocking out spinocerebellar pathways in the lateral medulla. And again, all those, the vestibular nuclei, spinocerebellar, descending sympathetic tract, and the trigeminal nerve, those are all ipsilateral to the lesion. The right-sided lateral medulla is corresponding to the right side of each of those features. The reason you get contralateral loss of sensation in the rest of the body is because you're knocking out the cuneate and gracile nuclei as well as the spinothalamic tracts, which have already decussated. And then as far as what vessel is causing that problem, so classically the posterior inferior cerebellar artery is what you would say on an exam, but in reality usually it's the vertebral arteries in about 80% of these cases, and that's what was happening with this lady. When she turned her head, it was somehow constricting the vertebral artery and cutting off blood supply to the lateral medulla, and giving her these sort of ischemic attacks when she turned her head to back up her truck. Okay, next problem. A 25-year-old man falls off the back of a truck and is brought into the ER. He's alert and oriented, but also drunk. All vital signs are stable. He states he cannot feel anything in his legs, but when you test with a pinprick, he can feel pain on his right side, but not the left side, just up to the level of his stomach. And conversely, he can feel you touching his leg on the left, but not on the right. You notice some small fasciculations or spasm in his right leg muscles, so that would suggest upper motor neuron injury, right? And when he tries to get out of bed, only the left side of his abdomen flexes, which suggests some sort of right-sided flaccid paralysis. So what is most likely wrong with the patient? Well, if we draw this out, which is always a good idea to do just to keep track of things, it's hard to keep track of all of that in your head. So drawing it out, you can see that there's some sort of flaccid paralysis and loss of sensation right at the level of the belly button, and then below that he seems to have some sort of spastic paralysis suggesting an upper motor neuron injury, as well as loss of touch and vibration on that side. The only thing that seems to be unusual on his left side is this loss of pain and temperature in his lower extremity. And this seems to correspond quite well to a classic syndrome called Brown-Séquard syndrome, or hemisection, meaning half of the spinal cord is injured. And this can happen with all sorts of injuries, whether it's falling off the truck and breaking the spine and having a piece of bone smash the spinal cord itself. Sometimes with stab wounds, the knife will only pierce half of the spinal cord, things like that. It creates this sort of classic syndrome that tends to pop up on board exams. And it's worth pointing out the reason here that there's not a loss of pain and temperature right at the level of the lesion is because the spinal thalamic tract, even though it does immediately decussate, as we say classically, the spinal thalamic tract carrying pain actually tends to go up one or two levels and then decussate and then ascend. So that kind of explains that unique little phenomenon there. But the reason that you have that blue bar, the loss of motor and sensory right at the level of the lesion, is because you've actually knocked out the nerve roots as well, the dorsal and sensory nerve root and the ventral motor root. All right, next problem. A 43-year-old woman states that her left arm and leg have begun feeling weak, and it's difficult for her to show her teeth on the left side of her face. Her right eye does not adduct, and the right pupil is larger than the left pupil, so a single lesion causing these effects is located where? So I put this problem in just because we didn't actually look at midbrain, but it is worth just kind of taking a quick note of. So if you look at a slice through the midbrain, you can see cranial nerve 3, which innervates the eye and explains these lesions that she's having. So not being able to adduct her eye and the right pupil being larger because you've lost the parasympathetics that come through the third cranial nerve. So she must have some sort of right-sided lesion of the third cranial nerve. And then along with that, it seems like she has some sort of cortical bulbar effect, meaning cortex to the face, which is part of the cortical spinal tract. The cortical spinal runs to the body, but when it specifically runs to the face, we'll call it the cortical bulbar tract. And you can see here that the cortical bulbar tract is sort of a lateral component of the cranial or of the cortical spinal tract. And so if you knock out this sort of anterior mediolateral location on the midbrain, you could knock out simultaneously the third nerve and cortical bulbar tract, which explains exactly what deficits the lady is experiencing in this case. And I also threw in just a nice anterior view of the brainstem here, so you can see the motor and sensory nuclei. For example, the trigeminal sensory nucleus, you can see how it runs so far vertically, or rostrally-caudal here. Then lastly, let's say a 24-year-old man has an aneurysm that causes a ptosis and a weakness in moving the eye up and down or inward, so again, adduction. At rest, the eye deviates outwardly, like an exotropia, and does not constrict to light. So where is the aneurysm? So kind of a two-stage problem here. We have to figure out the lesion and then figure out the sort of three-dimensional vasculature that would go around or supply or compress this area in the brain. So this again sounds like a third nerve palsy of some sort, with the loss of adduction and the inability to constrict, so a dilated pupil. So if we look at this image with the vasculature overlaid on the brainstem, things start to become more apparent. So even this image is not great at representing the three-dimensionality. Like for example, the carotids up at the top there, they're actually coming up vertically through the carotid canal and cavernous sinus there. So the angle between that internal carotid artery and that posterior communicating artery, the anastomosis there, that what's called the PCOM, posterior communicating artery, the angle between those two is actually much sharper than it looks like on this image. And it just so happens that the third nerve passes right in between that angle. So if there's an aneurysm at that junction, then it could easily compress the third cranial nerve. And that's what we're seeing here. So the answer would be at the junction of posterior communicating artery and internal carotid artery, D. Alright, so when we started, you guys hardly imagined that you could get familiar with histology, but I bet you're actually more familiar already with it than you would imagine. And I'm going to try and show you that here. So I'm going to give you this question that seems really difficult at first, but you actually know enough now to solve this problem. You're all going to be pathologists after this lecture. So a 40-year-old woman stops speaking mid-sentence and then has a seizure for the first time in her life. A CT scan shows a hypodense mass near the gray-white junction of the cortex at the left pars opercularum. And you biopsy it and send the sample to the pathologist, who sees the following image. So if you're shadowing in neurosurgery and you do one of these biopsies, you actually should go down to the pathologist and see how they prepare it. There's a couple different ways, which we'll talk about in a minute, but when you look under the microscope here, the important part is that you recognize, first of all, that these are structures that you've seen before, just in a different manifestation. So I would ask you, do you see several large neurons here? No, we don't. But what do we see? Well, we see small, round, dark nuclei. These are glial cell nuclei, and you probably recognize that. But you can clearly see that this is much more dense than what you saw on the earlier images. There's an abundance of glial cell nuclei filling the tissue here. And not only that, but on the left side you can see that there's even a couple little cell nuclei that have mitoses going on, and the nuclei are not all exactly the same. They're what we would call pleomorphic. There's a plurality of different morphologies to the nuclei. And in addition, there are several different densities. Some of them are super dark, some of them are kind of lighter stained, even though they're all with the hematoxylin, the purple. So all those nuclei are unusual. And if we look in the middle image, we see what's early vascular proliferation, what's called glomeruloid vascular proliferation. And then on the right, we can see it looks like all the cell nuclei are lined up together. So it almost looks like this parade of cells, what we would call palisading cells. But they're not actually palisading. That tends to be more with tumors and neurodevelopment of specific kinds of tumors, I should say. This is actually a pseudo-palisading. What's happening here is that the cells have gotten so dense that there's a central area of necrosis here. You can see several cell nuclei missing from the center of the image, and it creates this sort of pseudo-palisading, this pseudo-lining up of cells around the perimeter where they can get just enough oxygen and nutrients to survive. So, as you might imagine, this is a glial cell tumor. And there are several different types of glial cell tumors. Astrocytoma, grade 1, would be the least invasive, the least severe form of these glial cell tumors. But all of the things that we just talked about with the images that we see, that is indicative of the worst grade of glial cell tumor, which we would call a glioblastoma, or in other words, grade 4 astrocytoma. It's the worst grade of astrocytoma. So if you said C, glioblastoma, then you're right. You got this. And unfortunately, these are a very common tumor. And they manifest typically in this way. Typically in mid to late age, someone has a first-time seizure and gets a scan, and it shows some unusual lesion in the brain. Sometimes those can be metastases from other cancer sites, but unfortunately, it's really sad because these typically aren't found until they are grade 4, and by that time it's too late. These glial cell nuclei have basically migrated throughout the brain, so even if you resected this site, you couldn't get all of these glial cells. There's just no possible way. Even if there's one glial cell that's migrated to the opposite side of the brain, if you miss that one, it will proliferate. And so there's a lot of research going on about the genetics of these types of cancers, and there's still debate about optimal treatment regimens for this. You can give these little chemotherapy wafers at the lesion site after resection, but in this case, because the lesion is so close to the pars opercularum, which is Broca's area, it's the form of speech. In this case, it's probably better to try not to disturb this area of the brain, because regardless of what you do, this is a death sentence. Unfortunately, no one has survived glioblastoma. The average lifespan after diagnosis is typically 3 to 5 months, maybe longer. Some patients can go years even, but typically it's really short. There are some treatments, like I said, with chemotherapy or other resections, but the ultimate goal is to keep the patient as functional as possible for as long as possible, basically to keep the quality of life as high as possible. So if you went and tried to resect this, you could, most likely you would destroy their ability to speak, and that would drastically decrease their quality of life, typically measured by something called the Karnofsky score. And so in this case, it's probably better to leave it alone unless there's some sort of swelling and bulk effect. I'm actually really interested to see if CRISPR technologies will enable us to attack these sort of cancer cells specifically, and I'm also working on patient-derived stem cell models of these brain tumors. So you can take a patient's own cells and create these tumor models and see if there are targeted therapeutic regimens that will optimize treatments for these patients according to their genetics and their cancer type. It's also worth noting here that medulloblastoma down at G is the most common childhood brain cancer, and it's becoming one of the most lethal cancers, partly because the most common cancer, which is leukemia in children, the most common childhood cancer, is so well treated now that it just turns out that the second most common cancer, medulloblastoma, is taking over as the primary cause of death from cancer in children. Okay, let's move on to the very last section of this histology. So normally we would do this in a separate week. So this is the subsequent week's lab. So if you're exhausted, that's fine. Feel free to turn it off, take a break, come back next week. So this is an important part of understanding how we actually obtain the tissues that we're looking at. So it consists of these processes of fixing the tissue and sectioning it, then staining, and finally observing it. So for the tissue fixation, there's a couple ways of doing this. One is just to do the standard, put the tissue in formaldehyde and let it fixate. Remember, formaldehyde is that carbon double bonded to an oxygen, highly reactive, basically covalently bonds anything around it. So that's how it preserves the tissues. Now, if you have a tissue specimen from an animal that you want to keep carefully preserved, for example, something that you've genetically modified or that you've done some sort of important experiment on, you can sedate the animal that the tissue is coming from and inject formaldehyde directly into the heart. And in this manner, you will deliver formaldehyde to the tissues before you manipulate and remove them, and this preserves the structure more perfectly. But of course, you have to make sure that this is a necessary step and that you're sedating the animal so it feels no discomfort. And interestingly, there was actually a patient not too long ago that was, I think she was a lady in Russia who accidentally got a bag of formaldehyde and intravenously, and of course died quickly from that excruciating event. Now, once you extract the tissue and you have it preserved, then you can slice it in a couple different ways. If it's room temperature, one of the fastest ways of doing this is simply freezing the tissue and cutting it. But that's not perfect, and it causes some difficulties with staining later. So another way of doing this, especially if you're going to preserve portions of the tissue for a long period of time, is to immerse the tissue in wax. But the tissue is typically hydrophilic, so it's full of water, right? So we have to get rid of the water by washing it with ethanol. Ethanol is sort of that intermediate hydrophilic, lipophilic type molecule. It's the starting of a carbon chain, the two carbons. So it's kind of in between. It helps wash out the water and prepare the tissue for a lipophilic type substrate. So by washing the tissue with ethanol, higher and higher concentrations of ethanol, and then ultimately an organic agent like xylene, then when we put wax over the tissue, it should absorb quickly and permeate the tissue. This wax then helps support the tissue, and you can take the whole block of paraffin wax and put it on a microtome, which is this machine that moves the block of tissue up and down against a blade and slices off these thin sections, typically 8 to 10 microns, so a single cell thickness of your tissue sample. Then you can take this paraffin wax and float it on water and just simply put a glass slide under it and pick up the piece. These glass slides, they typically have a positive charge, and tissue tends to be negatively charged. So when you pick up the piece of tissue on the glass slide, they typically suction together and you can't really move or adjust the tissue without breaking it. So because the tissue is protected by the wax, these can typically store for quite a while, and this is how the tissues in our lab are stored for this course. And we'll see in a minute that in order to stain these tissues, we're going to have to actually remove that wax to continue the preparation process. But let's really quickly talk about another type of way to prepare these tissues, and that is with thin sections using a cryostat. So what we can do is take the tissue block, put it in this little plastic bin or any sort of holder, and fill it with this compound called OCT compound. OCT just stands for optimal cutting temperature compound. It doesn't really tell you what the compound is. It's actually like a vinyl alcohol, which is not good to get on your skin, just FYI, so don't touch it. Now, we put this clear solution of OCT compound around the tissue block, and you can also use it like a glue when it freezes. So when we put it into a cryostat, which is kept at like negative 20 degrees Celsius, this OCT compound will freeze and turn white all around the tissue block, and it actually holds the tissue block to this little round stage. The stage is what we put into the cryostat, and the machinery in there will move this stage against a blade and make slow progressions so that it slices off again about 8 to 10 microns at a time. You can then pick up these little thin slices of tissue that are frozen with just like a paintbrush from Home Depot or somewhere. You'll find that it actually depends a lot on the humidity of the day and the temperature. Like sometimes negative 18 degrees Celsius will make all the slices curl up, but negative 20 is perfect. Or maybe the humidity that day will make things different than the day before. So it's just kind of a funny behavior. But in essence, you pick up these little slices of tissue, you put them onto the positively charged surface of the glass slide, and then they just sort of slurp down and stick. So then you can take the slides and do any sort of staining process from there. So the slides that I would normally have you guys prepare, they were prepared in the wax, like I said. And so the first step is to take away the wax, usually with an organic solvent. In this case, we use Citrosolve. We try not to use benzene or toluene or things that are going to cause leukemias. But Citrosolve is a safe organic solvent that can wash the wax away. And then we bring it towards an aqueous environment by washing in 100% ethanol, then 95% ethanol, and then staining with a substance, in this case, Luxol-Fast Blue. So Luxol-Fast Blue is a stain that we purposely try to overstain, and then we're going to wash away the excess using a reagent, lithium carbonate. So the Luxol-Fast Blue, it sits overnight at 37 degrees Celsius. The warmth helps accelerate that reaction, helps it get into the tissues more, and then we wash that away with its own solvent, the 95% ethanol, and then finally bring that to a true aqueous environment by washing in water for 3 minutes. Now when you wash these, it's literally like coloring Easter eggs. You can just dip them in bins that you've pre-prepared for the proper reagents. So the Luxol-Fast Blue, we call it a regressive stain because we purposely overstain everything and then use a reagent to wash away the excess. The ethanol that we use after the lithium carbonate sort of quenches that reaction, and then we finally terminate that with an aqueous environment of water. And that prepares us for the next reagent, which is an aqueous reagent. Cresol violet needs to be in an aqueous solvent, and so we've done the first stain, gotten to our aqueous environment, and now we can stain with Cresol violet, which is a progressive stain. It will get darker and darker, and then we simply remove it from the staining media when we're satisfied with the results. So there's a lot of things here that have sort of been timed out for you guys, but obviously certain conditions can vary day to day and year to year, and so typically what I'll have you do is try to figure out what you would have done differently with your slides. If something goes disastrously wrong, you're not docked for it. You just have to say what you would do differently. So typically things that'll make a dye stain darker are higher concentration, longer time in the solution, a higher temperature, pretty common sense things. Whereas if the stain was too dark, then maybe you left it too long or too high of temperature or too high of concentration, or maybe you used too much reagent to wash out the stain and therefore you washed it all away. So just think about these things as you sort of go through these steps. And then after the Cresol violet, we just wash it out with its own solvent, water, and then we want to fixate the tissue and preserve it for long term. So if you have a frozen section, like we talked about with the cryostat, you would need to store that at like -80 degrees. There's -80 degree freezers, and they can store things for a long time without them degrading over time. But if you just stored it in a normal freezer, it would probably go bad. So the wax tissue is the wax preparation is one reason that we do that first option for these tissue preparations. And then once you've gone through the staining process, you can actually put it back into an organic solvent like Citrosolve that not only helps preserve the tissue against any sort of like bacterial contamination or degradation, but it also helps clear the tissue. It matches the refractive index of membranes of cells, for example, and it makes it so that when you look under microscopy, the tissue only shows the things that you've purposely stained, and everything else is clear. Otherwise, we would get a lot of noise and diffraction and blocking of light and things like that. So this final step, moving from the aqueous solution back to ethanol and then ultimately an organic solvent like Citrosolve, that helps clear the unstained tissue and preserve the tissue for long-term storage. And then we can take a mounting medium, essentially a glue, clear glue, and put it over the tissue and stick a cover slip made out of glass on top of it, and now our sample is perfectly preserved for long-term use. So when you go through that process, this image shows kind of what you should end up with. Nice purple Cresol violet stained neurons and nice Luxol-Fast stained blue white matter tracks. So again, I'm not going to score you on this part. You just get points for doing it. Make sure you go over the questions to consider at the end of the worksheet. It basically asks you questions about what you would do in certain scenarios if your stain was not turning out correctly, and then make some basic calculations of things, remembering that the easiest way to do these calculations is just conservation of mass equation, where C1V1 equals C2V2. Concentration times volume gives you the mass of a substance in a solution, and that mass will stay the same regardless of concentration and volume that you adjust. So the C1 times V1 equals C2 times V2. And also a useful thing to remember here is that when we talk about percent concentration, percent can mean a lot of things. It can mean a weight to weight, a volume to volume, a mass to volume, yada yada. So what we're talking about here, the standard definition of percent in most of biology and chemistry refers to an amount, a mass per volume. So when we're mixing a powder and we want something like a 0.5% solution, what you're aiming at there is X percent equals X grams per 100 milliliters of solvent. So that comes from the fact that a gram of water is a milliliter of volume. So if you had 1 gram of a powder in 100 milliliters of water total, then that's a 1% solution. You have 1 gram per 100 milliliters of solvent. So that's how you calculate percentages there. So that should help you with that portion. Again, you don't need to turn this in. It's for your own benefit, but these kind of questions do pop up on the exam. So that's why they're there. All right. You guys are awesome pathologists now. I hope that all made sense. Hope it wasn't too exhausting. Great job. Feel free to ask if you have any questions. Thanks.